Bismillah ar-Rahman ar-Rahim. Hello everyone. This is Solving Past Paper 2019, May-June 3-1, question 2. We're going to start solving question number 2 by making the outline drawing for the J1 slide, which is provided by Cambridge. Now, if you read the question carefully, you will see that the question asks you to draw an outline. I'm not going to show you the drawing. The question asks you to draw an outline for the slide after you view it by the microscope. I'm going to show you the slide, how it's going to look like. So here's the slide under the microscope. Now, as you can see, the slide is not stained. It's black and white only. You need to be able to distinguish the different areas in the slide. You need to distinguish the different tissues by looking at the size of the cells as well as the shape of the cells in the slide. Now, if you are a little bit familiar with the transverse section of roots, you will be able to somehow identify the different layers in this slide. As you can see right here, there is the epidermis. Let me show you the epidermis. So here's the epidermis. Underneath the epidermis, there is a layer which is called the collenchyma cells, which is somehow like this. After that, you're going to look at the parenchyma tissue layer, which locates right here all the way until you reach the endodermis. Now, underneath the endodermis, you're going to find the xylem vessels, those large xylem vessels, as well as you're going to see the pith. So let me show you how to draw this, the outline using the 100x magnification or 100 magnification in order to draw an outline for this drawing. Let's see some of the students' drawing. Let's start with Ahmed's drawing. Now you can take a look. So as you can see, here's the epidermis. It should be a little bit thinner, Ahmed, because you need to draw the layers in the right proportion. So I think for this layer it should be a little bit thinner. So he drew the epidermis by using two lines to show the thickness of the epidermis. Underneath the epidermis there is the there is the collenchyma layer, and then there is the parenchyma layer right here. Now, if you label Ahmed the cortex, where are you going to label the cortex? Can you label the cortex? Right, the cortex is basically the area between the endodermis and the epidermis. So can I borrow your pen, please? So the cortex, it goes from the endodermis all the way to the epidermis. So this is the cortex. This is the epidermal cells, the collenchyma cells, and here's the parenchyma cells. Now we can see underneath the endodermis, there is a layer of cells, and in that layer of cells you can see the xylem vessels. Now you might ask yourself, would I draw the xylem vessels or not? Sometimes. So again, you might wonder if you are going to draw the xylem vessels or not. Now I think in this specific specimen you need to draw the xylem vessels because they have a very few number, as well as because there are other types of tissues that locates in between the xylem vessels. So in order to show those other vessels, you need to distinguish between the two areas. Let me show you another slide for the same exact type of roots. By the way, this root is called the iris root. I'm going to show you another transverse section for this root. So here's another transverse section for the same type of root. It is stained. You can see the different layers clearly here because the different layers are stained by different colors. What I need you to look at is basically the xylem vessels, which locates in this type of tissue. Now, in order to distinguish between the xylem vessels and this type of tissue, you need to draw the xylem vessels by drawing circles in that area. So this is basically how you're gonna draw the xylem inside. But you need to make the wall a little bit thicker, Ranim. The wall of the xylem should be a little bit thicker to show that the xylem vessels have thick walls. Okay. Here's another example about drawing a plant diagram for another type of plant, and this is how you draw the xylem inside. So sometimes you need to draw individual cells, specifically xylem, in some areas to show that there are other tissues or there is other tissues around the xylem. That's regarding question number 2, the first part of question number 2, which is drawing an outline. The second part of this question 2, it has to do with the drawing 4 cells from the cortex. Now you should be familiar with the cortex. Now you need to pick 4 cells from the cortex, every one touching 2 other cells. So basically what you're going to draw after you view the cells under the microscope using the 400x magnification, you need to draw 4 cells. You need to make sure that the cell wall is represented by 2 lines, and whenever you touch 2 cells or whenever 2 cells touches each other, there is 1 line in common and there are other 2 lines. So the total number of lines would be 3 lines. In this drawing, you need to be aware that there are some air spaces in between the cells. You might not, it might not be easy to recognize them. You need maybe some experience to see that there are air spaces in between. So make sure that your drawing is big enough to fill at least half of the space provided by the examiner. I think, Ahmed, here is some mistake right here. Now, regarding this line right here, it should be fixed. Okay? Try to make clear drawing, and here's another mistake right here. This feathery line also should be, this one should be removed. That was the second part in this question. If you move on to the to branch B in question number 2, now you need to use this figure 2.1, and compare it with J1 slide. And you need to find out the differences between the J1 slide and figure 2.1 using a ruled line labeled by Q, R, and S. Okay, now regarding the last, regarding branch B in question number 2, you need to find out the differences between the transverse section in the exam paper and the J1 slide, which is provided by Cambridge. Now you need to find out what differences you can see between those 2 slides. Now Ahmed now is going to label the exam paper, this one. You need to label it and with some annotation about the differences between the J1 slide and the exam paper transverse section.
Difference in shape of xylem, ring shaped on J1.
So Ahmed is saying that the xylem vessels are distributed in X shape right here, while the xylem vessels right here are distributed in ring shape. So this is one of the differences. So you need to say J1 is distributed, the xylem in J1, Ahmed, the xylem in J1 is distributed in ring shape, while the xylem right on in the figure 2.1 distributed in X shape. Can you see other differences? Any more other differences, Sabaya? Any more other difference?
And there's no pith here.
If you look at the center of the transverse section, you're gonna see the pith region right here. But right here, you're gonna see no pith. Any other differences?
Width of cortex.
The width of the cortex. Now, the cortex in the figure 2.1 is much thinner compared to the width of the cortex in the J1 slide. So you can write that as another difference between those two specimens. Now, here's one more difference, which is the shape of the transverse section, the shape of the transverse section of the entire root. Now the entire root here is more into being regular circle, while right here it's not regular. Okay? This is regarding this part of the question. One more part remains, which is about the calculation. Can you please, Ahmed, maximize this file? Yes. Okay. The last question in this exam paper is about finding out the actual width of the root. Now, how can we find the actual width of the root? Now, as you can see, guys, here's a scale bar which is provided underneath the transverse section. This scale bar has a value, so you need to use a ruler in order to measure this scale bar. Scale bar, you need to use the paper itself, because there's going to be differences between this one on the board and the one on the paper. In the paper, I think it's 1.3 centimeters. So the scale bar is 1.3 centimeters.
Scale bar 1.2.
1.2? 1.2 centimeters. Can you please write centimeters? So 1.2 centimeters. Now you need to make a ratio between, now it is 1.2 centimeter, which is equivalent to 273 micrometer. Now you need to make a ratio between this Z line and the scale bar to find out the actual length. Can you show us, Ahmed, how you calculate it? So 10.3 divided by 1.2 centimeters to make a ratio between those two lines.
8.58
centimeters,
which means 8.58 pieces can fit into the line Z.
Mm-hmm. So 8.73 micrometer
multiplied by 8.58. 2342.43.
Now
whole number.
You need to round it as whole number because you are using the micrometer. Whenever you use micrometer, round it to whole number. Thank you, Ahmed. Thank you very much. And this is, yeah, write down the unit, please. Micrometer. Yeah, thank you very much. This one should be erased. Yeah, centimeters. Yeah. Sorry, my bad. My bad. Right here, this is a ratio. It has no unit. It's only a ratio between those two lines, and then use the value of the scale bar multiplied by the ratio. It gives you the actual size of the entire root. Okay. Any questions?
No.
Bye.